Oh my goodness. Oh my.
Okay, guys, so set remind when they don't have what is it? I don't know what you're saying. Can you like come send a reminder? Well, actually, I don't have my phone. But I'm teaching. I'll send it later. Maybe if they could watch it, they'd be here. I mean, if they, if they're awake.
Isn't that the point of life? So they can watch it like when this is
or later is what I was thinking.
I wish I would have watched this one here.
Or
this one at home. And then I'll put it in the conversations that are recorded.
All right, guys. So now here we go. So when we look at DNA replication, you have the so again, it's the new strand that's being built is going to be built from the 5 to the 3. So then, as the helicase unzips this, you kind of want to check like which direction. So our first enzyme that's going to be involved is Rna Primase, or just primase, and it's going to add RNA nucleotides. So it's going to build a little like section of RNA nucleotides, and that allows DNA polymerase 3 to come and then extend that and add on. Now this is where base pairing rules come into play. So when we learn like, oh, A, T, C, G, C, A, T, whatever, this would be in the original strand, and then this is what polymerase is attaching. So this is also where like mutations happen. When we talk about point mutations, like substitutions, silent mutations, nonsense mutations, missense mutations, this is where that is happening during replication. And so on the other strand, though, primase, because this strand on the bottom is going to be built in the opposite direction of the replication fork, DNA polymerase would then come and then extend it this way. So now, though, as helicase continues to unzip, when we look at this, I know I have DNA polymerase 3 had disappeared, but in reality, it's going to be building this continuously. Like it's just going to keep following DNA helicase. So we call this strand the leading strand, and it's built continuously in the same direction as the replication fork. Whereas the bottom strand here, now that more DNA has been exposed, now a new primer of RNA nucleotides will be added, and then DNA polymerase can come and, oops, that was fast, can come and do base pairing rules and extend it. Well, it's going to stop when it hits the other primer, right? It's already like a dead end right there. So now, helicase will keep on going. This DNA polymerase 3 will continue to base pair and will be following the helicase enzyme. Over here, you'll have another primase will come, build a new primer, and then you have DNA polymerase 3 will extend it. Now, our problem that we have here is that you have RNA still in that sequence. So there's going to be DNA polymerase 1, and DNA polymerase 1 is going to come and it's going to remove those RNA nucleotides and replace them with DNA nucleotides. Now, what you notice up here, though, is that it couldn't. It didn't add any nucleotides to this end because that's the 5' end, and DNA polymerase cannot attach to the 5' end. So here, you have this strand will get a little bit shorter at that end. And then you need to seal these Okazaki fragments, so there'll be the enzyme ligase that will build those phosphodiester bonds between those fragments. And then this will continue all along these 2 strands until you have 2 strands that are half original, which would be the black line, and half new, which would be the light blue line. So then here you have semi-conservative replication. Okay. So now, DNA and RNA molecules have structural differences that define function. Ooh, that defined function. So our genetics is in this double helix based on the base pairing rules, and we read those nucleotides in groups of 3 called codons, and that's our genetic information. But when we look at the structural differences, so here we have a double helix that makes it possible to like have mitosis or meiosis and pass on the genetic information. So that double helix allows it to separate and then build new complementary strands, and now you have 2 strands in each daughter cell, or 1 in each daughter cell. Versus RNA, RNA has lots of functions, and so RNA, you can have messenger RNA, where it's just transcribing or making a copy of a gene and traveling to the ribosome. You could look at tRNA, so being single-stranded, RNA can fold up into a tRNA molecule, and then RNA can also be a part of a ribosome. Ribosomes are made of RNA and protein. But then we also have small things like microRNAs. MicroRNAs are going to be used in protein production and regulating the proteins that cells make. And you have like small nuclear RNAs when we talk about spliceosomes. So RNA has lots of different functions, and that single-stranded shape allows it to fold up in different ways. And so when we look at our DNA, you can see, oh, it's in yellow, kind of hard to see, but you can see the fact that it opens up allows it to now create two daughters. Oh, this is making RNA, sorry. So this is making RNA in transcription. And then you have tRNA, and our RNA is your ribosome. Okay, so genetic information flows from a sequence of nucleotides in a gene to a sequence of amino acids in a protein. This is the central dogma of biology. So you have DNA. We're using RNA polymerase is going to make an mRNA copy of that gene. And then from that RNA, you read it in codons to get a protein. Okay, so that's our process of transcription and translation. I'm pretty sure we'll go into more detail with this. So when we look at RNA polymerase, when it's doing transcription, it's going to be only opening the DNA that is within that like transcription bubble. It doesn't totally unzip like a jacket or something. It's just that section that's being copied into RNA. And so another key point to make is that RNA is built from 5 to 3, just like DNA. So when deciding which strand to use, you need to think of where are the 5s and where are the 3s. So if your DNA, you have 2 strands, you have a strand like this of DNA, and then the anti-parallel strand of DNA, you have to pick the top or the bottom to be the template strand. You're going to say, okay, well, my RNA needs to be built from 5 to 3 if I'm going this way. So then I'm going to use the bottom strand is the one that will be the template strand. The top strand, then, here we would call the coding strand, the one that's not used. So pay attention to those 5s and 3s, and remember it's built. Both DNA and RNA are built that way. And then for us, our eukaryotes, once your DNA has been transcribed into mRNA, we do something called RNA processing, where we can edit our RNA. And so this is where we're going to have exons and introns, and we will actually splice out those introns and then splice together the exons. And we can edit this in different ways, and that's how you can have like 25 to 30,000 genes, but over 100,000 different proteins, depending on the different mRNA transcripts that we make. When we look at eukaryotes versus prokaryotes, you can see that prokaryotes do not do RNA processing. They don't have a nucleus. And so that's not right. But they don't have a nucleus either. And so when we look at this, there is actually a like physical separation between transcription and translation in eukaryotes, as well as a time. Like this is going to happen first and then second. Versus prokaryotes, they're going to have their transcription and translation happening simultaneously. So as RNA is being transcribed and formed, ribosomes will come and attach and then start translation and building that amino acid polypeptide chain. Okay, like without editing the RNA. Okay. Ooh, goodness gracious. So you start with DNA, you have replication, you get more DNA. If you have DNA, you can do transcription. Transcription is making RNA. RNA then can be translated into proteins, and those proteins could be enzymes or they could be structural proteins, and then enzymes are going to help with all of this stuff. Okay. Ooh, phenotypes, yo. You know what I'm realizing is not in this PowerPoint, and it might be later. What are the DNA experiments? They're supposed to be with the standards right here, and I don't know where they are. So let's go ahead and review the different experiments. Are you ready? Ooh, 1908, T.H. Morgan. T.H. Morgan is working with fruit flies. And so before Morgan, though, you have like Mendel, and Mendel's like, oh, factors are passed on from parents to offspring. What are these factors? What are these things? And then chromosomes are discovered, and then it's like, oh, chromosomes are made out of DNA and protein. And so T.H. Morgan is studying these flies and realizes, oh my gosh, these factors or these traits are passed on chromosomes from parents to offspring. And if you remember your chromosomes, if I take my penis, are made out of like DNA wrapped around proteins. The histone proteins, remember that? And so now T.H. Morgan is like, he actually wins the Nobel Prize in 1933 for figuring out that Traits are inherited on chromosomes. There can be sex-linked traits because notice the difference between males and females for some. And then he also did gene mapping, was able to map the chromosomes of the flies and figure out where each gene is located. Flies, the fruit flies have only 4 chromosomes, so it's kind of easier than with humans with 46. And so 1908, oh my gosh, traits are inherited on chromosomes. And then you skip over 20 years or so, we're now with Griffith. And Griffith is the one who's working with trying to find a cure for pneumonia. So with Griffith, he's the one who works with the mice. So with the mice, he infects the mice. Oh wait, it's not dead yet. You know, mice are one of the few things I can draw. I'm pretty good at penguins. Well, this is not a good one. Okay, so then he's working. So he's working with mice and to try and find the cure for pneumonia. Pneumonia is a bacterial infection, and so he works with bacteria. There's 2 different kinds. There's the harmless strain, which is rough under a petri dish, like when you look at it, or you have the smooth strain, and that one is the virulent or pathogenic. So if I were to draw these out, this is the one that doesn't work, right? If I were to draw these out, you have 1 and 2. So if you have the rough strain or the smooth strain, the reason why the smooth strain, this is the one that's pathogenic, because it has a capsule like around the top that helps it to like evade your immune system. So that's the difference like in phenotype between the rough and the smooth. So this one can be like attacked and defeated, where this one, this capsule helps it survive inside of you, and that's why it lives long enough to like cause disease. So when we look at what he was doing, he was infecting the mice. He infected the mice with rough strain, which is the harmless, doesn't cause disease strain, and the mouse lives. Then he infects mice with the pathogenic strain, the smooth strain that's covered in a capsule. Oops. And the mice die. And then he injects these mice, with heat-killed smooth strain ones. So it's dead bacteria that he's infecting this mouse with. And these mice, sorry, they live. Now this one, he takes this and adds them together, mixes them together, and infects these mice, and they die. So what the heck, right? So he's like, oh my gosh, something in this heat-killed one transformed this rough strain to become pathogenic, and it changed its phenotype. It transformed it to all of a sudden be pathogenic. Now back then they didn't know that it was actually bacteria picking up DNA from its environment. Now we know that's what's happening. But so Griffith was like, oh my gosh, there's something being passed on that can influence phenotype. And then we hop forward 16 years to 1944, where you have Avery, McCarty, and McLeod are like, well, what is the transforming factor? What is transforming the phenotype? And so they have narrowed it down to 3 options. They're like, well, you know what? It could be RNA. It could be DNA. It could be protein that is the transforming factor. Now, they were leading towards protein because when you look at the monomers of these molecules, RNA has 4 nucleotides, A, U, C, and G. DNA has 4 nucleotides, A, T, C, and G. But protein has 20 amino acids as its monomers. So when you look at all the variety of life, you would kind of hypothesize, you know, Protein is going to be that molecule of inheritance because it allows for so many more different combinations and so much more diversity. So they were kind of leaning towards protein as a molecule of inheritance. So what they did is they took and used enzymes to kind of isolate and purify each of these macromolecules. So in the bacteria, what they did is they used like proteases, RNases, and DNases that will like destroy, hydrolyze these macromolecules. So what they did is they mixed the bacteria with molecules that would destroy DNA and destroy protein to isolate just RNA. And did it transform? No. Then they took and they used enzymes that destroyed the RNA and the DNA, but left the protein okay. And then did it transform? No. And then when they left the DNA intact, and they mixed the bacteria with proteases that hydrolyzed proteins and molecules that destroyed RNA, enzymes that cut up RNA, leaving only DNA, yeah, then it was transformed. The bacteria went from rough and non-pathogenic and was transformed to smooth and pathogenic. So they were able to confirm, oh my gosh, DNA is the molecule of inheritance. But then people were like, are you sure? Let's check it out in viruses. So that's where you have Hershey and Chase. And Hershey and Chase are like, well, let's see if viruses also do the same thing. So you have bacteria, then you have these cute little bacteriophage viruses. And these bacteriophage viruses have a protein coat, but they also have DNA. They also have DNA. Oh. They also have DNA inside. Oh my God. I think it might be time for new markers. Okay. So now you have DNA inside. So they were like, well, how does this virus infect the bacteria? Does it use its DNA to pass on the information about building new viruses, or does it use protein to pass on the information? Now, lucky for us, when you look at molecules and how they're built, DNA is made of C, H, O, N, and P. What? Protein is just C, H, O, and N. So what they did is they made this DNA with radioactive phosphorus because it's only located in DNA, not protein. Now, some amino acids, though, have sulfur as part of their R group, the little like variable chains that hang off of each amino acid. So sulfur is not found in DNA. So they made these protein coats with radioactive sulfur. So now they can kind of follow what ends up inside the bacteria. Is it going to be sulfur that's inside, or is it going to be phosphorus that's inside? And so what they did is they allowed the viruses to infect the bacteria, and then they used like a blender. Oh my gosh. And they spun it. And what's going to happen is the heavier stuff is going to settle down to the bottom. So the bacteria will settle down to the bottom. And when they did that, down here in the pellet, they found lots of phosphorus, in radioactive phosphorus, and they found sulfur like floating more near the top. So that tells us that, oh my gosh, it was actually the DNA that entered into the bacteria, not the protein coat. So again, they were able to show that DNA is carrying genetic information, not protein. And then the question becomes, okay, well, DNA is now the molecule of inheritance, but what does it look like? What is its structure? So then you have people working on this. It was actually a race amongst scientists. And so you have Chargaff. Chargaff was working on DNA, and he's the one who, by looking at different species of organisms from fungus, bacteria, plants, animals, like all different like domains, came to the conclusion that, oh my gosh, no matter what organism you look at, the number of A's equals roughly the number of T's. There might be some variation, maybe not exact because of mutations, but the like A and T and C and G go together. Now it's unique for different organisms. So like in humans, we might be like 19.8% A and 19.9% T. And so that together, about 40% of our DNA is A and T, let's pretend. But a dog might be 25 and 25%. So it's the percents of A's and T's is different in each species, but A and T go together. And so their percents are pretty similar, and C and G percents are similar. So this was like a piece to the puzzle, and that helped scientists know, okay, so we have these nucleotides, and somehow they're pairing together. And so some of the first models of DNA structure, I think Pauling, I think he built like a triple helix. And then some other ones had the nitrogen bases on the outside and the sugars and phosphates on the inside. So there's a couple different hypotheses and that kind of stuff. And that's where you now come to like Rosalind, Franklin, and Watson and Crick. And so with that, Rosalind was taking photographs using X-rays. And so they have photo 51. And when you look at the X-ray of DNA, so if you have your double helix, here, so you have a double helix. And as you're like taking an X-ray, it's shooting the beam of X-rays this way. So as it bounces off is the imprint that it's leaving. And so by looking at the structure that's formed, you see an imprint and you can measure the twists of the double helix. And it's it's diameter. I'm not really sure the right word. And so now by looking at the photo 51, you could infer, oh, DNA is a double helix. There's this much spacing between turns, etc. And so when Watson and Crick saw the photo, they were like, oh, bam, it's a double helix. Built the structure, and now they get the credit, right? And so with that is how we get the structure. But then you lead to, oh my gosh, but now we know it's a double helix. But how does it replicate? How do you get from a double helix and then two dotted cells also have identical copies of DNA, like during mitosis? And this is Messelson and Stahl's semi-conservative replication experiment, where they use the heavy and the light nitrogen. Okay, so with that, let's go ahead and look at, so what they did is they started with DNA that was in the little bacteria and E. coli that were made of heavier nitrogens. So you start out with DNA that has N15, which is a heavier nitrogen. So if they were to centrifuge this in a little tube, it would like kind of settle lower. And so they allowed these bacteria, though, to replicate. But they only gave them access to nucleotides. Oh. So a nucleotide is a 5-carbon sugar. So only gave them to nucleotides where the bases were N14. We're a little bit lighter. So let's go ahead and do that in blue so we can follow it. So here, we'll make these new nucleotides N14. So as the bacteria replicated, they have their DNA is going to open. And now the new complementary strands that were built by leading and lagging overlapping fragments and all those enzymes are going to be half N15, half N14. So when you look at like the little centrifuge tube, it's a little bit higher. Oh, it's right in the middle. It's half old, half new. Then as these open up, so then here's the red, here's the blue. And so now these strands have opened, and as they allow the F2 generation, you're going to see this. Okay. And now when you look at the little test tube, you're going to have two lines, the N14 and then about N14 and a half. And this is how Messelson and Stahl showed that DNA replicates semi-conservatively. The most beautiful experiment in biology. And then now we know how it replicates, but how do you read it? How does this actually code for life? There's only 4 bases. And there's like billions of them. And that's where you had like the, there was actually a group of male scientists called the RNA Club. And they had green ties with gold thread of the RNA. And they spent years trying to decode it. And I think it was Crick. But one of them realized, oh, it's read in groups of 3. They were kind of disappointed that it was so simple. And so that's where you have like the genetic code. Okay. So that's your experiments. And actually, do we need to review transcription and translation real fast? Maybe a little bit. So when we look at protein synthesis, the central dogma of biology, what we have here is you're going to have your, when we look at, here's DNA, and then we're going to open up a section of the DNA. And we're going to use one strand. We'll call this the template strand. And that template strand is what's going to be used to make RNA. And then this process is called transcription. And later in the PowerPoint, we'll talk more about transcription factors and regulation and that kind of stuff. And then here you have, once you get the RNA, we will edit it. There's RNA processing that happens. So in RNA processing, this is where you add a 5' cap, a poly A tail, and you cut out introns. And then you get your mature mRNA. And when it finds a ribosome, whether it's on the brachiar or in the cytoplasm, here you have a ribosome. And the ribosome is going to read it in groups of 3. And so those groups of 3s are called codons. And a codon might be like AUG, and that's going to code for an amino acid. In this case, it's methionine. And so if this first codon is AUG, you're going to have a tRNA. A tRNA will come and bring an amino acid. And then as our ribosome will move down, you'll build your growing polypeptide chain. And this process here is translation. So let's see what standards keep showing up. I haven't looked at the PowerPoint since last year. So I'm pretty sure it'll show up again. And if it doesn't, we can go back to this if we need to. Okay. So you have transcription and translation, and yeah. Okay. But I think we're moving on to phenotype text. So phenotypes are determined through protein activities. We really want to emphasize that your phenotype is based on protein. So while DNA has like all of your genetic information, your phenotype and what's actually expressed is an accumulation of proteins that are being made inside of you. So your skin color, that's melanin, that's a protein that's accumulating. Your eye color, if you have brown eyes, green eyes, blue eyes, that's protein in your eyes. If you have different genetic diseases, that's going to be the presence or absence of proteins. So when we talk about like let's just use being albino, for example. And so with this, if a person is, there's like 3 different options, right? So you could have like 2 make melanin or make pigment. You could have 2 chromosomes. So we have 2 copies, 1 from each parent. So let's say like here dominant is saying like makes melanin or pigments. And so a person could be heterozygous or homozygous recessive. So in this recessive allele here, no pigment is made. So pigment is a protein, so there could be like a nonsense mutation or something that created a not working gene. So that therefore when you went through transcription and then translation, there's no pigment made. But hey, if you have 1 chromosome that has a dominant allele, you're gonna make pigment just fine. You have 1 that is making it. However, if you are homozygous recessive, both copies are not working, so now you have the absence of that protein, and therefore your phenotype shows up as albino. Okay. And so this is in cystic fibrosis. And so when we look at this, in cystic fibrosis, there's a chloride ion channel. When we talk about cell membranes, how proteins are part of the cell membrane that allow ions to pass through because they can't, because of nonpolar core normally. So they have to go through a protein channel. Since your protein channels are coded for by your DNA, if you're missing a protein channel, that could cause lots of issues. So in cystic fibrosis, they have a defective or absent protein. And so now the chloride ions can't go through, and that influences osmosis. And so now the water doesn't flow. And so therefore you have this like a mucus accumulates. And so it's like a sticky mucus because there's not water going in by osmosis. And so there's all these breathing treatments and things you could do. But this is an inherited disease based on defective or absent proteins. So same thing, if you are a carrier for cystic fibrosis, you are fine because you have 1 allele that's going to code for the proteins and be okay. However, if you inherit 2 copies, this is going to be like you don't have any information on how to build that chloride ion channel. So like, but then diseases can also be, it's not always recessive like this. You can also have dominant issues. So like how I get those blood clots. So the blood clot, like protein, like I have more of that protein than an average person. So therefore my blood clot's more than the average person. So here I actually am heterozygous. So while the majority of you in this room is homozygous recessive, about 5% of you are actually heterozygous, by the way. You just don't know it. And so here, instead of it being a defective protein, I just make a certain protein that clots my blood. So I, as a heterozygote, am 7 times more likely to get blood clots than the average person because I have more of a particular protein. However, if I was homozygous dominant, I'd be 80 times more likely because I'd have 2 copies of the chromosomes, both causing this protein to be made. So it doesn't always have to be a recessive thing. Extra proteins can also cause harm. Okay. So here is what I was talking about with cystic fibrosis. So a normal like protein channel for that chloride ion. If the chloride ion comes in, well, now you have a greater amount of solutes. And so like the way osmosis will work. Whereas here, if it's not working, going out. Here, if the chloride ions can't leave, well then that affects water balance. Okay. All right. Oh, your eye color. I love this. So when we talk about brown eyes or blue eyes, it's the amount of protein that you accumulate. So a person with brown eyes actually has more of this protein in it, and therefore the way the light hits the eye is like kind of different. The way it reflects and is absorbed. So people with like lighter eyes or like blue eyes or something, they have slightly less of this protein, so the way the light bounces off is different, giving it a lighter appearance. So, good to me. Okay, genetic engineering. Genetic engineering techniques can manipulate the heritable information of DNA and in special cases RNA. So when you look at genetic engineering, one of the examples that we did in class is bacterial base transformation. Hey, what's transformation? Where the bacteria takes up DNA from its environment. So with Griffith, with the mice, we saw that the bacteria took up DNA from their environment. And so with that, you have the bacteria, as they take up DNA from their environment, that DNA has new genes in it. Then if those genes are expressed, the phenotype of the bacteria will now be different. When we look at variation in eukaryotes in us occurs by meiosis with independent assortment, crossing over, and random fertilization, as well as mutations. But in bacteria, because they reproduce by binary fission, that's like making identical clones. So their variation, their variation is based on mutations, but also the three ways that they acquire variation, one of them is transformation. They can pick up new genes from their environment. You also have transduction. When we talked about those phages, so in transduction, you would have this phage is like transferring, when the phage comes in and replicates, it can accidentally take up some bacterial DNA and then go pass it to a new bacteria. So that's another way. And then you have conjugation, which is with the sex phyllis, where they send out that like tube and the DNA can be exchanged that way. So transformation is one of the ways that bacteria do acquire new variation that's different than just mutations. But here, we're taking advantage of this. We're taking advantage of the idea that bacteria do take up DNA from their environment. So this is us like manipulating or making this happen. So here in plasmid-based transformation, you would take a plasmid. A plasmid is like a circular piece of DNA that's separate from their main chromosome. So when you look at a bacterium, when you look at bacteria, you have the, you have, so when you look at bacteria, let's try one more time, here we go. Okay, so you have your the bacteria has their main circular chromosome, but they also have extra pieces of DNA called plasmids that have maybe like 50 to 500 genes on them. So there's extra plasmids in the bacteria. So scientists, what they'll do is they'll take out the plasmids and they can genetically engineer these plasmids. And they do that with these enzymes called restriction enzymes. So if you remember with restriction enzymes, this is where they cut at specific sequences. So like EcoR1 cuts at like GAATTC or something. I don't know if I have that memorized right or not. But they cut them to produce sticky ends. Do I have a picture of that nexus? So here, when you cut with the sequence of the DNA with a restriction enzyme, you get sticky ends. But if you cut DNA from different sources with the same restriction enzyme, the sticky ends are all going to match. And because of base pairing rules, then the sticky ends can recombine together. So when we talk about plasmid-based transformation, scientists can take a plasmid from a bacteria. They can also genetically engineer it to be antibiotic resistant. And that's important. We'll talk about as we keep going. So they'll take a plasmid that is antibiotic resistant, and they'll cut it with a restriction enzyme. And whatever gene or DNA they're interested in studying, they'll cut that with the same restriction enzyme. And then they allow them to mix. They allow them to mix, and hopefully they recombine and form recombinant DNA. So then you have a plasmid that has a piece of human DNA or human or other source, non-bacterial DNA, as part of that plasmid. Then they'll like mix these bacteria with certain chemicals that kind of like loosen up the cell membrane. So now it makes it easier to take in that plasmid. When we did our lab, you guys got some bacteria. We used E. coli. We had plasmids in little tiny like tubes, and we took and we got the plasmid. We mixed it with the E. coli. We put it in different temperatures and added some like different chemicals. And so we did what we were adding chemicals and changing the temperatures, etc. We were trying to like loosen the cell membranes so our little E. coli would take up those plasmids we added. And so we had them take up the plasmids and then we put them on petri dishes and we left them off in an incubator. So not all of us had successful transformation though. Some of us had glowing bacteria and some of us didn't. So what was happening in that experiment, what we did is we grew bacteria that had, we tried to have them take up plasmids. The plasmids were already genetically engineered before we got them. We started out with plasmids that were antibiotic resistant. So when we poured, do I have a slide for this? Maybe? Oh, we can review here. I might have a slide on antibiotic resistance. So here we take a plasmid, and we have, let's say, human DNA. We're going to cut them with the same restriction enzyme. So that restriction enzyme cuts at a specific restriction site. And so now this enzyme will then cut, producing sticky ends. And there we go. Okay. And then we'll cut the plasmid with the same restriction enzyme. And so now you have the plasmids kind of been opened up. And so then hopefully we form recombinant DNA. And so now this plasmid has a piece of DNA from a different source. So recombinant DNA is DNA from 2 different sources. So now we have a recombinant plasmid. And then we're going to hope that the bacterium takes up that plasmid. And that part right there is the transformation aspect. And so now the RNA polymerase will read that gene and then transcribe it into RNA. And then the ribosome can read it and produce a protein. In our little experiment, the protein that's produced was a glowing one, so we could literally see it happening. So with this, was that too fast? Okay. And so, if it's insulin or any other thing. So if you have bacteria, this is an antibiotic-sensitive bacteria, meaning it'll die in the presence of antibiotics. Now, if the scientists, though, take plasmids that have been genetically engineered to not be killed with antibiotics, then when they insert the plasmids, or hopefully the bacteria take the plasmids up, if the bacteria successfully was transformed or successfully took in plasmids, then if you grow it on a petri dish that has antibiotics in it, only the bacteria that have that plasmid will survive. And this is how you can isolate. So if you have like a whole little like tube with a bunch of like millions of E. coli and you mix plasmids, how are you going to find the ones that actually took up your plasmid? Is by using antibiotic resistance. Because only the bacteria that took up the plasmid will survive in that new environment. So therefore, you now have which ones have transformation, and the ones who didn't die. Now, if you are working like you chopped up DNA with restriction enzymes, and you don't know which one has your gene of interest, there's other steps you can follow. For us, we used a gene that was glowing. And so what we did actually in ours, if you remember operons, which operons will come up in our PowerPoint later, but bacteria, they can turn on or off their gene expression using operons. So what we did is we added to that, or I did before your lab, is I added the inducer molecule into the Petri dish. So that Petri dish had the inducer molecule, so the bacteria could turn on gene expression and glow. But without that inducer molecule, they didn't. And that's why you had 2 plates that had antibiotics. And you may have had colonies, but one would be glowing and one wouldn't be glowing. It's because the absence or presence of an inducer molecule to turn on gene expression. So we did both plasmid-based transformation and gene expression in one lab. What? Okay. So your College Board standards mention 2 techniques. In the one little thing that says genetic engineering techniques, they mention plasmid-based transformation, PCR, and electrophoresis are the 3 that they like specifically to talk about. So in PCR is if you want to amplify and make lots of copies of a section of DNA. And so with that, you start with your target sequence. And then you denature it with heat. Ooh, but DNA relies on enzymes for replication. So we have to use special enzymes from, the little archaea that live in hot springs. So we're going to take the enzymes from little, like, prokaryotes that live in, like, super hot environments naturally. So their enzymes don't denature in hot temperatures. So we're going to take Taq polymerase from like an extremophile that lives in hot temperatures, an archaea, and we're going to use that as our DNA polymerase, like to add the nucleotides. So in PCR, though, there's a couple things that DNA needs for DNA replication. You have to add nucleotides. You have to add the ends like a primase to build the primers. Actually, do they use it? I don't know if they use primase. The primers might just attach. Huh, interesting. Okay, then you need to add primers. And then The enzymes. So the way it works in PCR, you're going to heat up the DNA and it separates, it denatures, it breaks those hydrogen bonds between the A and T and the C and G. The A and T have 2 hydrogen bonds. The C and G have 3 hydrogen bonds. And if we didn't talk about, I don't know how specific College Board will be, but just to refresh your memory, when we talk about A and T, C and G, those nitrogen bases, that's where nitrogen is found in your DNA, CHON, and the P is in the sugar phosphate backbones. And that phosphate has a negative charge. Okay. But the A and G are both purines, and the C and T are both pyrimidines. And that is based on like their ring structure. And so with that, like I think purines, A and G, have a double ring structure, and pyrimidines have a single ring structure. I could be wrong. I don't like to memorize things. So, but there you go, just in case. So anyway, you're going to add, you denature it though, and then the hydrogen bonds break. And so now you have 2 strands, and then you kind of cool it down, and then the primers will anneal to the opposite sides, or like to the sides of where the, depending on like 5 to 3. And then you have the enzyme Taq polymerase will then add new nucleotides based on base pairing rules, and now build the complementary strands in semi-conservative replication. And then you heat it up again, and those separate. And then you cool it down, the primers anneal, and then Taq polymerase will extend and make new copies. And then you heat it up again. You do like in like 30 times of this, you have over 1 billion pieces of DNA, billion copies of your target sequence. And so it's basically exponential growth of DNA replication of just a small sequence that you're interested in studying. So that's PCR. And then you have, oh, here we go. You denature your DNA, heat it up, and then annealing, you cool it down a little bit. Primers will attach. And then you have the extension part, which is where the Taq polymerase enzyme will come and build the complementary strands. The other thing that College Board mentions is gel electrophoresis. And so with this is where if you take a piece of DNA and you chop it up with restriction enzymes. Restriction enzymes cut short sequences. Like an EcoR1 is like GAA-TTC. If my DNA is 3.2 billion base pairs, GAA-TTC is going to show up frequently because it's such a short sequence. And so here with this, you like have a restriction enzyme is going to cut the DNA up, and then it cuts it into different size fragments. Now, gel electrophoresis works on 2 things, charge and size. So then you're going to take these DNA fragments and put them like into one of these sample wells. So each well, it's like a little hole in the gel, will be a different sample of DNA that you want to compare. So then you'll put the DNA fragments into one of these, and then you turn it on for about 24 hours, and the pieces of DNA will separate out based on size, but it's also because of the charge. So DNA has a negative charge because the sugar phosphate backbone. You're climbing a ladder of DNA that your hands are holding onto. So that phosphate has the negative charge. So you have a whole bunch of negatively charged molecules, and at this side of the gel, you have the negative electrode. It's going to repel, and it's going to be attracted towards the positive side. Now, if you only let it run for like 24 hours, the DNA pieces are going to move, but the ones that are going to move the fastest are the smallest. So therefore, you get like a banding pattern with the smallest pieces of DNA fragments moving the furthest. Okay. Yes.
But it's cutting up the same sequence every time. How are they different types?
Because it's well, for me, mine will always look the same. But like me and you are going to be different. Yeah. So it's like mine will always have the same pattern. So in the old, like now we have more advanced technology. So in the old days, you could use this for like a crime scene or for paternity tests because mine will always be the same. But then if you think about, well, how would humans, if we're all the same species, have so different? Well, if you look at our DNA, guys, our DNA is like only 1% is actually your genes that codes for your phenotype. But you have so much like junk DNA that doesn't code for anything on your phenotype. So if mutations happen in your junk DNA between genes, they're not selected for or against. Those mutations are just happening, but they're not coding. They're never in our phenotypes. So they're kind of, I don't say they don't matter, but they're not like going to be selected for or against. So therefore, you can have unique differences because we have mutations happening in not important pieces of our DNA. But that part might be important, and we haven't discovered it yet. Like for a long time, we thought, oh, just junk DNA. But now we find out that some of that non-coding DNA codes for microRNAs used in protein production regulation. So we're like learning more about DNA as time goes on. So here's like an example. You can see like different banding patterns. Like this one here would be for the same individual. Okay. Ooh, illustrative examples of products of genetic engineering include genetically modified foods, transgenic animals, cloned animals, pharmaceuticals such as human insulin or growth factor X. So, for example, I think in class I use insulin a lot as our example. So with like insulin, little bacteria can like produce insulin like crazy for us. Little tiny factories, because it's the universal code of life. Like the genetic code is the same for all. So like if I have a gene sequence like AUG, CCU, etc., I can read that. My ribosomes can then translate it into a protein, but so can a bacteria. However, we would have to make sure that we already give the bacteria, like when we genetically engineer it to make our proteins for us, we've given them the like final copy of the final copy of the gene, because they don't have introns. So they're not going to, or they don't have the machinery to cut out introns and splice and process RNA. So if you really wanted bacteria to produce a human protein, you'd have to give them an edited piece where the introns are already cut out. Does that make sense? Okay. So here though with cloning, we didn't really talk about cloning in class, but here's how it works. So one of the first sheep first animals cloned was Dolly. Bless you. And so what they do is they can take an egg from a sheep and they can take off the nucleus, because the nucleus is only haploid, right? And so they can take a donor cell where that nucleus, they'd be taking like a somatic cell, like an ear cell or something like that, that already has both sets of DNA, one from mom, one from the dad, already a diploid. And so what they'll do is they'll remove the nucleus from the egg and then get the nucleus from the donor cell into the egg and then allow the egg to start to divide. And so then by mitosis, so now you have like an embryo that has the original, like the donor DNA, and there was no sperm involved. There was no fertilization. It was just like, here's a complete nucleus, now start dividing. So the first time they cloned the sheep though, they didn't really have a clear understanding of epigenetics and how like our genes can be altered like based on environmental like issues or experiences. So Dolly the sheep aged really fast and died of old sheep diseases at a young age because they didn't clear off the epigenetic markers on the donor DNA. So, okay. Oh, cell cycle. What? Okay. So the cell cycle is a complex set of stages that is highly regulated with checkpoints, which determine the ultimate fate of the cell. So when we look at the cell cycle, there are different checkpoints. They're going to make sure the cell's healthy and good to divide. So in that G1, this is where the cell is living its life. The G1 checkpoint is going to make sure that this is a healthy cell that's going to divide. But a cell's going to spend most of its life in G1. This is where cells like living, making proteins, etc. If a cell receives a signal to divide, so that would be like a growth factor or something. So it could be like a platelet-derived growth factor. Let's say I get a cut, and so now I'm like bleeding. Well, then I'm going to have platelets are going to come, and the platelets are going to form a scab. Well, now the platelets are going to send out signals called platelet-derived growth factors. These growth factors are external signals that will attach to like pretend like a receptor tyrosine kinase and start like a signal or like a cell response within the cell to then have the cell start to divide. Ooh, we have a cut. Now that ties into like our immune system. So with our immune system, we need to like repair this. This is an area where now intruders and bacteria and pathogens can get in. We need to start to heal this by mitosis and repair that damage. So therefore, the cell will begin mitosis. So that would be an external signal saying it's time to divide. But we only want to like divide healthy cells. And so therefore the G1 checkpoint is going to check for DNA damage. And if the DNA is damaged, they can repair it possibly, or they can send the cell to like cell death, apoptosis. And so with that, it's kind of like cells, like there's actually like a suicide cascade where the proteins that the signal transduction pathway that happens results in the cell like dying. And then like going through blebbing and folding up, and then their scavenger cells will like part of your immune system, like macrophages will come and clean up that debris. So if you have damaged DNA, the cell shouldn't divide. Now, if it does and it runs that stop sign, this is one of the ways that like cancer can develop when like damaged DNA starts to replicate, and now you have multiple cells with damaged DNA. So the G1 checkpoint It's going to check to make sure that there's no DNA damage, that there's nutrients, etc. And if the cell is cool and healthy, well, then it can go into S phase. And S phase is where DNA replication happens, where now you have your primase, your helicase, your Okazaki fragments, your ligase, etc. And that's where you have semi-conservative replication happening in S phase. Because now the only reason a cell would ever have to make copies of its DNA is if it's dividing. Because in the cell cycle, you're going to have interphase where you have your DNA, your chromosomes that are linear chromosomes, and then it receives a signal to divide. Oh my gosh, we got to make 2 identical cells. You have to copy all of your DNA. So this is where you're going to take one strand of DNA, you're going to copy, and you're going to make 2. But they're temporarily attached by a centromere. And this is where you have your sister chromatids, which are identical copies of DNA. And then later during M phase, you're going to pull these apart, and each daughter cell will get an identical copy of DNA. Okay. So you have checkpoints within the cell, and then inside the cell is also regulated with internal molecules like MPF and cyclin-dependent kinases. So a kinase is an enzyme that phosphorylates proteins and makes proteins active. And so here, cyclin-dependent kinases are constant in your cell. But what varies, depending on where you are in the cell cycle, is the amount of cyclin. So once the cell receives a signal to divide and it's going into S phase, the amount of cyclin is going to increase, and that's going to attach to CDKs, activating them, which then activates other proteins involved in cell division. The cyclin-dependent kinases are going to help the centromeres stay together, the sister chromatids. They're going to be involved in activating enzymes that break down the nuclear envelope. All the steps involved are going to be, not all the steps, but a lot of the steps involved are going to be regulated by these cyclin-dependent kinases, internal molecules that help push the cell into M phase. Okay. Ooh, so here's your cyclin and your cyclin-dependent kinases. So cyclin-dependent kinases stay constant in the cell, but it's cyclin that varies. And when cyclin attaches to the CDK, you get MPF, M phase promoting factor. And so that'll help, once MPF is in high enough concentrations, that'll push the cell into M phase and allow it to pass that G2 checkpoint. And then that'll be involved in when the nuclear envelope breaks down, you have the spindle fibers, all of those steps involved in mitosis. And then eventually, though, that cyclin is degraded. And that cyclin will be degraded right around the same time, whoop, the sister chromatids get pulled apart. Okay. So when we look at mitosis is going to pass a complete genome from the parent cell to the daughter cells. Mitosis is used for growth. So when you are a baby, you're really firstborn, the sperm swims into the egg. Oh my gosh, you're one cell big. So now that cell has all the directions to build you, but it's just one cell. And today, here you are, trillions of cells later, all made by mitosis, all with the exact same DNA, but just different genes being expressed. So every single cell in your body has the exact same DNA from that first cell that was fertilized, the egg and the sperm. And now, when it divides, it went to 2, and then to 4, and then to 8, 16, 32, 64, etc. And now, as the cells are dividing, this is where development comes into play. And we turn on different genes and different cells that will determine their fate. So we use transcription factors to regulate which genes are being expressed. And that guides our development and our cell specialization. So today we have 200 different cell types, but they all have the exact same DNA, but each cell has different transcription factors and different genes turned on. Okay. So, but it is, in mitosis, it passes a complete genome from parent cell to daughter cell. And so when you start with one cell and you go through, sister chromatids, okay. And then they get pulled apart. Now you have 2 identical diploid cells. Oh, look at that. So pretty. Hey, what stage is this? Is this the beginning? Actually, College Board, you don't have to memorize prophase, metaphase, anaphase, telophase. But you can identify what's happening. So here in this is in the very beginning of M phase. So here, guys, if you see this, here's the nucleus. You can see how it's just kind of blurry for the blue stuff, right? This is chromatin. This is your DNA uncondensed. It's not until right at the beginning of M phase that your DNA actually coils up. So it's kind of like if you were to take your hair, I don't know, and you twist it and you twist it, it gets shorter and shorter and shorter like this. So that's kind of what's happening. You have these long strands of DNA, but it coils up and it gets highly condensed, where you can actually see the chromosomes underneath the microscope. So that's what's happening here. Like you can see individual chromosomes. Otherwise, in interphase is G1, S, and G2. M phase is the time when it's actually dividing. So what we're looking at right here is M phase. So here the chromosomes become visible. And then because we want each daughter cell to get an identical, let's see. It doesn't say a complete genome, but identical daughter cells. We want to make sure that these sister chromatids get evenly separated. So here they all line up in the middle of the cell, and that's metaphase, but you don't even know that. And so here they're lining up along the middle of the cell. Hey, what are these red things? Spindle fibers, great. And then here in this top one, the sister chromatids are getting pulled apart. And now each daughter cell is going to have an exact copy of the DNA. And then you'll have cytokinesis happen. Cytokinesis where you have the cytoplasm starts to fold in. It forms a cleavage spur that kind of looks like a butt crack. And then eventually it'll separate and you have two daughter cells. Can a plant do cytokinesis? No. So instead, a plant is going to build a new cell wall. I don't know if it's like that. It's going to build a new cell wall, and then it'll like separate it like that. Okay. So that here is the end, and you can see the new cell membrane between those two cells. Meiosis. Meiosis, a reproduction division. Followed by fertilization, ensures genetic diversity in sexually reproducing organisms. So with meiosis, here we go. In meiosis, do you want me to put my meiosis PowerPoint? Or do you want to just go like how we've been doing? I really like my meiosis PowerPoint. Can I switch over real fast? Yeah. Okay, so let's review meiosis.
I need to get coffee next time. Huh? I need to get coffee next time. It's my third second. For bio.
How you guys doing? All right, guys, so here we go with meiosis. So with meiosis, this has our 46 chromosomes. So here you have the 46 chromosomes that are in chromatin. This will be what it looks like in interphase. So what's going to happen here, when the cell receives a signal to divide, they actually will condense. So I literally do have 46 chromosomes here. I copy and pasted and counted. And this is what 46 looks like. But to learn meiosis using all 46 would be, like in humans, would be kind of complicated. So I'm going to use less chromosomes. So here, to refresh your memory on some of this vocabulary, when we look at like gametes, meiosis is making gametes, which are sperm and eggs, are sex cells. And they're made of meiosis. And so in humans, each of our gametes have 23 chromosomes. So when, we call them haploids, they have just one set. So when a sperm swims into the egg, or fertilizes the egg, here we have 46. So now this is fertilization. We have 46 chromosomes total. Now we're back to diploid. So when the standard talks about restores the diploid number, that's what it's talking about. If we reproduced, if we made gametes by mitosis, our number would constantly increase, like exponentially. If you, made an egg with 46 and a sperm with 46, well, then you'd have 92. And if they made a sperm with 2 and an egg with 92, like, no, it doesn't work like that. But do organisms reproduce asexually? Yes. Yeah, so you can have some organisms who actually will reproduce by mitosis, but then their offspring are all clones. And the only differences would be due to mutations because mitosis makes identical daughter cells. But the benefit evolutionarily of sexual reproduction is that there's variation created. So we have mutations create variation, but so does sexual reproduction. Because during meiosis, you're going to have crossing over, which we'll see. You're going to have independent assortment, and you're going to have random fertilization. So evolutionarily, sexual reproduction is favored or selected for in a lot of populations because of the variation. Because now you have variation that's been created, and now allows greater chances of survival in changing environments. Natural selection. What? Okay. Here we go. So with this, now the diploid has 2 sets of chromosomes. One from the mom and one from the dad. This is why you have 2 copies of each allele. But once that fertilized egg, that zygote is formed, From that point on, the cells reproduce by mitosis. To make a hook, I hate it. And so with this, the only time you would do meiosis is in your reproductive organs, in your ovaries or in your testicles. That's the only place meiosis is happening. Meiosis happens in your ovaries. Okay. Or mitosis happens in your ovaries. So now other words though, we talk about haploid as being one set of chromosomes. We represent this with an N. And so here you have N plus N is 2N. So when we say a diploid number in humans, 2N equals 46. And then N equals 23 for us. In fruit flies, there, I told you earlier they had 4 chromosomes. So in fruit flies, 2N would equal 4. And then N is 2. 2 plus 2 is 4. That was a little tricky when we went outside with the pool noodles. You guys remember that? How everyone was like a little unsure? Okay. Here we go. Okay. So now, when we talk about karyotypes and chromosomes, so here is when we talk, a karyotype is like a picture of an individual's chromosomes laid out from biggest to smallest. And here you have a haploid organism. I have one of each one. So when I say, oh, you have one from mom and one from your dad, you have 1 through 22 plus an X or a Y, 1 through 22 plus an X or a Y. So with this, here we have, this would be all the chromosomes in an egg. So when the sperm fertilizes the egg and it brings the second set of chromosomes, now you have a diploid organism. You have 2 of each one. This is fantastic in terms of helping us survive like genetic mutations that are negative. So we talked earlier about like cystic fibrosis. If you inherit, let's say in the egg, that like on chromosomes, I don't really know, but pretend on 14 right here is the mutation that says cystic fibrosis. Well, guess what? You don't have to have cystic fibrosis. If the next allele you get is dominant and it works, hey, you're fine. You're heterozygote. You're not going to have that disease because you're expressing that dominant allele. So by being a diploid, that's kind of helpful. If you do get a chromosome that has a negative mutation, hopefully your second one is working and functioning. Does that make sense, kind of? Now, when do we not see this, though? In males. So in males, if they receive like a negative like allele, like a harmful allele on their X, they don't get a second shot. They don't have a second chance. So like hemophilia is a disease inherited on the X chromosome. That's the opposite of me, where I make too much of that protein and I clot my blood. In hemophilia, they don't make that protein and they don't make scabs. So they bleed to death. And that's an X-linked or sex-linked inherited disorder. It shows up more frequently in males because if they inherit a recessive allele, they don't have a second shot of another X to have the dominant working allele. You okay with that? Okay. So this would be diploid. One from the mom and one from the dad. Okay, here we go. This is called a karyotype. And so we look at these chromosomes are called homologous chromosomes because they have similar genes. So on this one earlier, I talked about cystic fibrosis. So one might say, oh, recessive, you have an absent or mutated protein. And then on this one, maybe working. They don't have to be the same. It doesn't mean exact. It means they have the similar traits on these chromosomes. So when we look at this, okay. It's not, this picture is not this. These are not sister chromatids. These are not duplicated chromosomes. That's just a pet peeve of mine. Okay. Because they're like crooked. You see that looks bad? It looks like an X. So this is what it would look like in sister chromatids. Sorry. Duplicated versus unduplicated. Okay. Oh, look at this chromosome from the mom. Look at this chromosome from the dad. They have similar traits. Eye color, hair color, height. They don't have to be identical, but they have similar traits. So we call these homologous chromosomes or like matching chromosomes. So here, when you look at this, let's go ahead and follow how this diploid cell that has 2 of each chromosome can divide and make gametes that have just one set. So with this, here, in interphase, here's your cell, 2n equals blank here. 6. That's okay. Don't be shy. So here in G1, then we go into S phase. And in S phase, they're going to duplicate. Okay, at the end, we should have n equals 3. Each gamete should have 3 chromosomes. Will those gametes be identical? No. Okay, so now here, when we look at this S phase, you're going to have the DNA is going to duplicate. The gametes won't be identical. Crossing over, independent assortment. Okay, so now here, S phase. Now in the beginning of M phase, so we go through G2, prepares for division. One of the first steps in M phase is going to be the homologous pairs are going to pair up. They're going to find their matches. So with this, they find their matches, and now, does this happen in mitosis? No. Like, if you want to know about the reason, like, why does this happen, Miss Neimeyer? Well, next crossing over is going to occur and create variation. But they pair up, guys, because the whole purpose is to get from 6 to 3. So by finding the matches, like here's one, here's another one, they come together, well, then eventually we're going to pull them apart. So each gamete gets just one. So by like finding their pairs, that helps to get organized to make sure that when they do separate, each gamete just gets one copy. They don't get 2 number 2s and no number 4s or something like that. Okay, so here, this we call a tetrad because tetra is 4. So here you have like 1, 2, 3, 4. And so we have a tetrad, which is made of 2 homologous chromosomes. And this is where crossing over is going to occur, where they're going to exchange pieces of DNA. It actually forms a synapsis, and they like kind of like zip together. It's like protein stabilizing them. Oh, I turned the air on. And so it's more than just one place. This is just showing you one location, but it really is quite a bit. Okay. So here, the chromatids are going to exchange pieces of their DNA. This is one source of variation in in sexually reproducing populations. And so here you have variation. Oh, a new combination that's never existed before. So pretty. Okay. And so then they separate. Oh, all right. And then here we go. So that's our recombinant chromosome. And then you have crossing over, though, happens in multiple locations. The areas where they cross over is chiasmata. So that would be like those areas. Okay. And the yellow highlighted there. Here's my karyotype from when I did 23andMe. So you can see the different colors in these chromosomes are different regions of the world where my ancestors came from. And so you can see like this, the yellow here is actually Native American ancestry. And you can see it's on different chromosomes. So it's like from crossing over, it's been like put on the different pieces. Also, when I looked at my children, one of my kids has 0.5% Native American, the other kid has 0. So when we talk about like how independent assortment, I think it's Rocco who doesn't have any Native American in him. And so that means when I have homologous chromosomes, so if I look at like, let's pretend like this one right here has a little bit of yellow. So when these separated in independent assortment, like when they get pulled apart, Rocco got this one and not that one. And like up here with this big piece of yellow, that means that Rocco got this one and not that one. So then it's random. And so I have only 2 children, and they have different amounts of Native American DNA, all because of independent assortment. Every time chromosomes move to the middle cell, how they get separated is different. And so kind of rad. Okay. So when we look at this, now we're going to go into the middle of the cell. So now we need to like get organized to separate our chromosomes and break it down from 2 end to end. So what's going to happen next? The homologous pairs, the tetrads that have already crossed over are going to move to the middle of the cell. But how they move to the middle of the cell is different. Every time they move, in us, we have 23 pairs moving to the middle of the cell. Sometimes this one will be on this side and this one will be here. That's going to be variation when they eventually separate and get pulled apart. And that's how my 2 kids have different combinations of my ancestry. Okay? And so now here they're going to get pulled apart. A whole lot of stuff. They're going to get pulled apart. The homologous pairs, the matching chromosomes get separated. So now this is the end of meiosis 1. There's 2 meiosis. Meiosis 1, meiosis 2. 2 rounds of division. Oh, cytokinesis. Are these cells haploid or are they diploid right here? Haploid. They have 3 chromosomes. But what's the problem? There's too much DNA. They're still in sister chromatid form. So then the next thing that's going to happen, if these are going to get separate, they're going to move to the middle of the cell. This is what mitosis looks like. And then these will get pulled, the sister chromatids will get pulled apart to opposite sides of the cell. And then cytopieces. And now I have 4 genetically different haploid cells. How are they genetically different? Well, crossing over and independent assortment. But what? Okay. In humans, there are 8 million different ways our chromosomes can align to the middle of the cell. Also, my sweet students, later when we talk about linked genes and genes being inherited independently from each other, So there's our two options. So when I look at these chromosomes, these genes here are considered to be linked because they're very close together and most likely going to be inherited together. Genes that are not linked would be like this one and like this one. They're on different chromosomes. There's different possibilities, different ways they can end up in gametes. And so also, sometimes genes that are really far apart on the same chromosome can also be considered not linked. So like this gene and this gene here, even though they're on the same chromosome, you can see crossing over separated them. So if crossing over happens more than 50% of the time between two genes, you consider those ones not linked. Because they're so far apart, you can't really predict that they're going to be inherited together because they're like crossing over is going to separate them basically. Okay. Oh, and then do we add tails? Oh, we do. Okay. Sperm. What would this picture look like if this was eggs? How many cells would I have here? Yeah, so there would still be 4 cells that are all genetically different, but 3 would be very tiny and 1 would be very large. Okay. All right. So let's switch back over to the other PowerPoint. To give you a heads up, it's 9:30 already. So we'll have a break in at 10. We'll take a little short break so you can stretch and that kind of stuff. Oh, we got to pass around the sign-in sheet too, guys. Oh, really? On slide 37? Okay. Adam Spaghetti of 3, we saw Spaghetti of 4. I guess speed it up, but this is the harder one, right? Yeah. Okay, guys, so meiosis is, so when we look at this, ensures genetic diversity. So we have random fertilization. We have crossing over, independent assortment.
I'm pretty sure maybe you tried them before.
Hey guys, if you could put your time in, if you already know you're going to be here till noon, put your time out too. And so one more time, because if you don't, I have to go in and fill it in. I have to remember who was here. Okay. All right. So here we go. This is just showing what we just talked about. Okay. Ooh, genetics. Now we move into genetics. The fun part of today.
Are you sure about that?
Okay, so it says the rules of probability can be applied to analyze passage of single gene traits from parents to offspring. So this standard is talking about just single gene traits. These are not dealing with like polygenic is a trait that is not a single gene trait. So for example, polygenic is a trait that is based on multiple genes, polygenic. So if I draw on the board a couple of different chromosomes, hey, does anyone at their tables have working markers? Do you guys have markers at work?
Yeah. Oh.
Hey, why did I draw 2? Would there be? I think that's 2. Okay. So let's pretend like here that all of these traits, I have 2 here, one, I think I have enough, yeah, okay. So you have multiple genes on different chromosomes, maybe in the same chromosome, but the idea here that whatever combination you get, like if all 3 of these genes, their protein products that they make are going to influence like your skin color, for example. So your skin color is a polygenic trait, meaning that you have quite a few different factors all at play that will determine your overall trait of skin color. That's one trait, one phenotype, but based on many genes. Versus something else like cystic fibrosis is a single gene trait. You either have like the allele that gives you working chloride ion channels or you don't. That's a single gene, not multiple gene products all influencing that trait. So this standard is talking about single gene traits from parents to offspring, and you can use probability. So when we talk about Punnett squares are just a tool to help you estimate probability. So with this, here we're looking at chickens. And so here with this, the probability is inside, well, let me start over. when you set up a Punnett square, like what you're really saying is what gametes could this parent make? So for example, I really sincerely am heterozygous for eye color. So on my genes, let's use this chromosome here, this set. So my eye color, if big B is brown eyes and lower B, little b recessive is blue eyes. Let's pretend. So I really am heterozygous. So during meiosis, we have Mendel's law of segregation, where my alleles are going to separate. And this is the whole idea of going from diploid to haploid. So each of my gametes should get at least one allele for eye color. If they don't, that's non-disjunction, and that's errors and mistakes during meiosis. And so here, all of my gametes, these alleles are going to separate during meiosis. I'll have 50% of my gametes will inherit a dominant allele, 50% of my gametes will inherit a recessive allele. So what I'm doing at the top of a Punnett square, that's what I'm writing. There's a 50% chance they'll inherit a dominant allele, 50% chance of a recessive allele. These are your eggs and sperm on the outsides of the Punnett squares. These are saying these are the possibilities that could happen in the gametes. And so the Punnett square is saying if these two parents get together, what could their children be? And that is the inside of the Punnett square, the possibilities. It doesn't mean if they have 4 kids, they're going to look like this. Now, in this example, this is not regular Mendelian genetics. This example is showing us non-Mendelian genetics. Sorry, I don't know what that's saying. This is going to be incomplete dominance. So incomplete dominance, where you have the homozygous dominant, the homozygous recessive, and then the heterozygotes have a different phenotype that's somewhere in between the two parents. Now, this is just made up from Google Images. But if this was a Mendelian genetics, what color would this one be? It would look like this, right? You'd have a 3-to-1 ratio. So when we talk about like ratios and stuff, this is where it ties in. So with the rules of probability here, we're talking about, I guess, well, what College Board may ask you is they could give you like numbers and stuff, and then you would figure out, oh, that's a 3-to-1 ratio. Those traits must be inherited as dominant or recessive. Like you fall into ratios and stuff and figure out what like the expected is. So here, just to refresh your memory, because it might make your life easier when you are looking at numbers and stuff in Punnett squares, is that when you have like a homozygous dominant individual, all of their offspring are going to have that trait. It doesn't even matter what the second allele is from the other parent. So if you ever see 100% of offspring having a trait, that in regular Mendelian genetics, that means that they're all at least heterozygous. They've all inherited one dominant allele. Now, if you have a situation like this, where you have a heterozygote is crossed with a homozygous recessive, your ratio is going to be a 1-to-1. So if you ever see numbers where half have a phenotype and half have a phenotype, a different one, you're like, oh, that's not 1-to-1. Wow, one parent must be heterozygous and the other parent is homozygous recessive to get those kind of numbers of offspring. And then if you get like something that's approximately a 3-to-1, that means that both parents were heterozygous. So you have to look at the numbers to figure that out. And so here, College Board will give you like little like word problems or something where they might tell you the numbers of offspring. Then you have to think back to, oh, what kind of parents would create that? Also, College Board may use words like a test cross. Oh, this, I don't know, pea plant was test crossed. If something is test crossed, that means it's crossed with a homozygous recessive individual. A test cross is always homozygous recessive. Okay, so they use that word kind of a lot. So if we like review vocabulary from genetics, you have words like the parental generation. The parental generation are the original parents, and they're crossed. Crossed means this. Someone earlier this week said times. No, mating, babies. Okay, and so your parental generation is first, and then their offspring. So here would be the F1 generation. And then if these were to mate, you see this in plants kind of a lot. In plants, they can self-fertilize. And so even though a plant is self-fertilizing, it's not mitosis. It's not going to be clones because they still made their sperm and their eggs by meiosis. There's still going to be variation. It's still random fertilization. And so the plant can self-fertilize a lot of times, and so that will be your F2 generation. Because if you see in plants, oh, the F1 were allowed to self-fertilize. That means they still did all of meiosis and made gametes by, made gametes, period. And then they fertilized. And so this is also where, sometimes if there are large chromosomal mutations in a plant and they self-fertilize, sometimes they can only end up reproducing with themselves because their chromosomes won't, have homologous pairs with others. And this is how you can have sympatric speciation happen. Because here their gametes are only working with their own. And so therefore you have a new species now because it can't mate and make viable fertile offspring with others nearby. So you see a lot of polyploidy, which means more sets of chromosomes, or sympatric speciation occurring in plants, because if they have these mutations that prevent them from mating with another plant, they can at least make their own offspring. Okay. So here, sometimes though you might see situations where you never get maybe homozygous dominant ones. You get a 2-to-1 ratio. In that situation, you have lethal dominance, where 2 copies of this allele, of a dominant allele, result in death. And so that would be, if you ever see a 2-to-1 ratio, that's what it's kind of implying. That combination of alleles is so deadly, they never form. It's selected against. Okay. Pedigrees, yo. Okay. Sometimes I actually don't really like when they have lines down the middle. I feel like that's cheating. So with this, do we know this is telling us, let's pretend the females weren't covered in. They weren't colored in, like these half-bloods. Could you tell what kind of let's talk about modes of inheritance, shall we? So when we talk about traits being passed on, can I back up just a few slides? So when we talk about traits being passed on, and we're looking at, oh my gosh, where did that, the other path, remember the karyotype? That's what I was looking for. So when we talk about chromosomes, we have autosomal chromosomes and we have sex-linked chromosomes. In humans, for example, 1 through 22 are your autosomal chromosomes. You have 2 of each one, and they're not related to sex at all. And so 1 through 22 are going to be your autosomal chromosomes, and then you have your X and your Y, or your X and your X, which are your sex chromosomes. So when we talk about modes of inheritance, how are traits inherited? Modes of inheritance. You have 2 options. You have autosomal. They can be inherited on an autosomal chromosome, and if it is autosomal, that means you don't have to worry about males having just 1 copy. Autosomal is when they tell you it's autosomal, they are meaning that you get you have 2 copies because you have 2 of each kind of 1 through 22. And now if it says sex-linked, that's males are going to have just 1 copy. Now things are going to be different in the ratios. So they're either going to tell you it's autosomally inherited or sex-linked. And then the second component to this is it's autosomal dominant or autosomal recessive. Sex-linked dominant, sex-linked recessive. So with hemophilia earlier, where the person doesn't make scabs, that's inherited sex-linked recessive. Versus brown eyes is inherited autosomal dominant. Or cystic fibrosis. Cystic fibrosis is when you are homozygous recessive. I can just add a little, where did I do with all my markers? I could just add CF for cystic fibrosis. That means you have 2 copies of a mutated version of the gene where your protein doesn't work or it's absent. That would be inherited autosomal recessive. Not all diseases are autosomal recessive. My blood clotting thing is autosomal dominant because I have the gene that makes a protein that causes me harm. So sometimes you have autosomal dominant diseases, like Huntington's disease is another autosomal dominant disease, but it doesn't become activated in your body until you've already reproduced. Because a lot of times autosomal dominant diseases, especially real serious ones, will most likely have the offspring die before they reach reproductive age, so they can be selected against in a population. And so if you look at the Huntington's disease, though, it doesn't be expressed until between 30 and 50 years old. You've already reproduced at that point. You've already passed on your dominant, you have a 50-50 shot of passing on your dominant allele. And so therefore, that isn't selected against in a population because it's not occurring before offspring. So it doesn't affect the fitness. If you remember, fitness is based on survive and reproduce. So people with Huntington's disease have already reproduced by the time the disease takes effect. So that's why it's a dominant disease that isn't being selected against. There's another disease that is autosomal recessive that's super deadly, is the one Tay-Sachs. I only talked about it in a few class periods, I think. Tay-Sachs is a disease. Let's just use this T right here. We can use a little T. In Tay-Sachs, in the brain cells, so here you have like, oh, that's really bad. So here you have like a neuron. There's your axon. Inside a neuron, in your brain cells, you have lysosomes. If you remember, lysosomes are the organelles that are kind of like the garbage trucks. And that's your nucleus. And so the lysosomes have hydrolytic enzymes where they break bonds by hydrolysis. Well, in people with Tay-Sachs, there's a mutation. So let's pretend like the A, T, C, G, A, T, C, C, I don't know, G, A, etc. They have one mutation, a point mutation, that now they don't make an enzyme that hydrolyzes lipids. They don't make an enzyme in their lysosomes, in the brain cells, all because of one point mutation, where now that enzyme is not made, and they can't digest fat. So when that baby is born, they're born fine and healthy. But then as their fat accumulates inside their brain cells, because they don't have the enzyme to hydrolyze it, the brain fills up with fat. They're blind. They're paralyzed by 1 year old. They eventually, the whole body shuts down by like 3, 4, or 5 years old, and they end up dying. All because of one enzyme is missing in the brain. All because of one point mutation out of 3.2 billion base pairs, one difference. And so that would be an example of a disease that is greatly selected against. Okay? And here we go. So modes of inheritance, my wonderful students. You have two options for looking at a pedigree. You could talk about it. You might also have mitochondrial DNA. Non-nuclear inheritance, though. What? Okay. So let's look at this. If you don't have the females colored in, could you tell what kind of inheritance this is? If it's only males colored in, you're going to go with sex-linked because it's showing that there's a disproportionate amount of males being affected by this trait. So therefore, if you see a pedigree with lots of males and not really that many females affected, go with sex-linked. Okay, like just straight up pick sex-linked because that's probably what college board is like implying. And then if you have like even number of males and females, you can kind of rule out sex-linked, kind of. You don't have to look for other clues. But when we look at this here, do you know how this trait is inherited? Oh, and by the way, when I say how is this trait inherited, it doesn't even matter the trait. It's just whatever, like whoever's colored in is saying they are expressing that trait. So now, is this trait going to be a dominant trait? No. No, you're like, well, how would they know that? Right? So you cannot, this is like saying, let's pretend you think, well, maybe it's autosomal dominant, Ms. Neimeyer. Okay. So then here, if it was autosomal dominant, then that's like saying that a blue-eyed parent and a blue-eyed parent had brown-eyed children. That's not possible unless a mutation happens, okay? And so here, though, could a brown-eyed parent and a brown-eyed parent have blue-eyed children? Yeah. And when we look at this, so this here, that could also happen in that one. So we can say kind of safely that this is an autosomal recessive trait. In this one, what do you think? There's one like pairing that lets us like eliminate a lot of our choices. Okay, so it's this one right here that will tell us, okay, it's autosomal. Sorry, that it's recessive. Because here, this is like saying like two dominant parents have a recessive child. And that is like this Punnett square here. You're like, what is she talking about? It's like if you have a heterozygote and a heterozygote, they can have a recessive child. Dominant can have recessive. That's what I keep talking about. Versus if I had like two, like only little a's, there would be impossible to get a dominant allele unless a mutation happens. So now here though, but if you're like, but Ms. Neimeyer, how do I know if it's autosomal or sex-linked? Well, if this was a recessive trait, which this pairing between these two parents tells us it's recessive. because it's not expressed in the parents, but it's in the offspring. That's like the main clue. Look for that first in your pedigrees. Like that's the first thing you want to look for. Then you want to say, because that will help to determine if it's dominant or recessive. Dominant or recessive. And then you're going to think to yourself, okay, but now is it sex-linked or is it autosomal? So then you look for other clues. Well, here I see this mother. She has a trait. And so if it's a recessive trait and this female expresses it, that means that in her chromosomes, it's this. So now, when she has offspring, her sons inherit her X. So therefore, any son she has will have that trait and express it. And we look here, and this son does not have it, does not express it. So that's how we know it's not sex-linked. So this is an autosomal recessive trait. Okay. So the clues you look for, you want to look at, are there more males than females? If there's more males than females, and if there's more males than females, but there are some females, the dad would have to have had it, because the daughter gets an X from the dad too. Is that okay? Okay, so if there's more males than females, that's a sign that it possibly is sex-linked. And then if it's even numbers of males and females, probably autosomal. But like you can always do process of elimination to confirm that. And then you want to look for this. Always look for this. If that shows up, then you can kind of confirm it's a recessive trait. If that's not there, you're just going to have to do look at other things. But usually, people who are testing you on pedigrees are going to be kind of kind and make obvious crosses that will show. Sometimes you could. What would this look like though if this was a mitochondrial mutation? What if it was a pedigree of mitochondrial DNA? What would that look like? It would only be passed from mothers to their offspring. So for example, in this, if it was mitochondrial, this mother would have it, and then all of her children would, but only her daughters would pass it on. Like it wouldn't be passed on here. Like that would not be colored in. Okay? Because your mitochondria come from your mother because the cytoplasm is so big, and the sperm just pretty much contributes DNA. All right. Oh, good job, guys. Segregation and independent assortment of chromosomes result in genetic variation. These are Mendel's two laws of inheritance. So I don't mind messing up. So when we think about Mendel, can we talk about Mendel right now real fast? So Mendel got real lucky choosing pea plants as his model organism to study inheritance. So Mendel, when we look at Mendelian genetics or non-Mendelian genetics, those are our two options of what we talk about in terms of how things are inherited. Mendel got super lucky because pea plants with their 7 characteristics, the peas were either green or yellow. The plants were either tall or short. The flowers were either white or purple. The pods were either constricted or smooth. The flowers came off the side or the top. Everything was one or the other. And if you see humans, when we look at our variation, it's not all just one or the other. There's so many ranges. So skin color is not is not Mendelian. It's not one or the other. There's multiple genes involved and giving us our phenotype. Mendel did not study traits that were polygenic. Mendel did not study traits that were incomplete dominance. Mendel did not study multiple alleles. Everything he worked with was one or the other. There was 2 options, tall or short, green or yellow. So we call that Mendelian genetics. Simple, genetics of how you learn in 9th grade, basically. And so here, Mendel figured out, without knowing about chromosomes, without knowing about DNA, he figured out that each individual has 2 copies. Has 2 copies of each factor. He didn't even know what genes were and just called them factors, which was a fancy way of saying things. Some things are passed on. So now Mendel is okay. You probably didn't do that. It's so anyway, so Mendel figured out, though, by following pea plants over generations, hundreds of them, not really thousands, but lots of pea plants, that, oh my gosh, the alleles separate. The information that gives the traits separate when gametes are formed. So he figured out that individuals are diploid and end up as making haploid gametes. He didn't use those words, but he figured that out. Mendel's law of segregation. Alleles separate. Hey, you're on your conversation's on YouTube, by the way, because we're live streaming on that service. Okay. So now here you're going to have Mendel's law of segregation. And then he also figured out that genes are inherited independently. So flower color, purple versus white flowers, has no impact on green or yellow peas. They were inherited independently on different chromosomes. So when we will talk about like Mendelian genetics and Mendel's law of independent assortment, when you do one of those big dihybrid crosses, like when you do this kind, here you are basically doing Mendel's two laws. So here, if you have an individual that is like heterozygous for two traits and you're doing a big dihybrid cross, like this, when you set up the top, you're basically saying independent assortment and Mendel's law of segregation, that these traits are not linked together. They're going to be inherited independently, and here's all the different combinations. So when you do this, you're saying like, okay, when this individual makes gametes, so if I look at the chromosomes, they can have like an A, they're heterozygous, and then the Bs. If they're on separate chromosomes, they can end up in different gametes, independent of each other. So you could end up an A and a B. If I divide this, we'll end up in a gamete together. I mean, a dominant and a dominant, not an A and a B, they're all A's and B's. Or the little A and little B could end up in gametes together. However, another possibility, though, what if the B's were switched because of independent assortment every time? So I can have the recessive be on this side and the dominant be on this side. Well, now I can have a dominant A allele could end up in a gamete with a recessive B allele. And on this side, I could have a dominant A allele and a recessive A allele and a dominant B allele. So when you are setting up your dihybrid cross, that is what you're saying. Here's the different gametes that are possible. So if you tie this into chi-squared and linked genes, so then sometimes if I were to test cross this individual and I cross them like this, well then every single gamete that they make will all be, because, oops, look, just kidding. Oh, that's all they have are recessive alleles. So every single gamete is going to be this in that individual. So here, all I really have to do is worry about this top row. And so here, you get like a 1:1:1 ratio. All of these have a 25% probability of being born because inside here are the different offspring. So when you do when you get data in a problem of something, let's say that these were fruit flies, and here there's 1,000 flies. And you would expect 250 of this phenotype, 250 of this one, and this one. But oh my gosh, Miss Neimeyer, that's not the results. The results I got look something like this. Let's say there's like 461 of this phenotype, and then, I don't know, 472 of this phenotype. So right here I have 39, and then maybe, is that like 28? So now here's what I actually got. Oh my gosh, this is when I would do a chi-squared. Are these traits really inherited independently? Because this looks kind of fishy. My observed is so far away from my expected. So then I would do a chi-squared to test the null hypothesis that these traits are inherited independently. And so when I would do a chi-squared, it would look something like this. I would take my observed, 461, minus my expected of 250, square it, divided by my expected, plus my observed minus my expected squared over my expected, and I would keep on doing that for all four. I'll tell you straight up, you're going to reject your null hypothesis. There is a difference. This variation is way far away from what we'd expect. When you see data like this is saying that these traits are not inherited independently. These traits are inherited on linked genes. I'm sorry, they are linked genes. They're inherited on the same chromosome. They go together. And when I look at the offspring, I see that this one looks a lot like this parent, and a lot of them look like this parent. And so the ones that don't are the results of crossing over. But what? Okay. All right, guys, it's 10 o'clock. So we'll let's go with like a 12-minute break because it's going to take you a while to get back in here. We probably won't start till 10:20. So go like to 10:12, and then we will come back and start at 10:20. Oh, how do I stop? Are you sure you stream?